Today I'm going to be talking about engineered immune cells, what they are, how we modified them, and how these technologies are being used to treat human diseases, particularly cancer. What is an engineered immune cell? Engineered immune cells are immune cells that scientists or clinicians have deliberately modified to perform enhanced or entirely new functions. These modifications can improve the cell's ability to recognize specific targets, increase their cytotoxicity, or fine-tune the signals that they respond to. Essentially, we're reprogramming the components of the immune system to behave in a controlled and therapeutically beneficial way. So why do we start to engineer the immune system? One of the main motives for this approach is the fact that cancer is remarkably skilled at evading the immune system. Tumors often downregulate antigen presentation, secrete immunosuppressive factors, and build a microenvironment that physically limits immune cell infiltration. Engineered immune cells are designed to overcome these barriers. They function as a living drug, and once infused, they can expand, circulate, and actively search for malignant cells throughout the body. There are many types of immune cells that we can engineer, but T cells are the most commonly engineered immune cells, especially in the context of CAR T therapy. Natural killer cells are also being explored because of their ability to kill cancerous or infected cells. However, more recently, researchers have also begun engineering macrophages because they are better suited for infiltrating solid tumors and remodeling the tumor microenvironment. In this presentation, I will be talking about CAR-T cells because they're the most widely known example. The CAR-T cell stands for chimeric antigen receptor T cell therapy. A CAR is a synthetic receptor that is introduced into a patient's T cells, as shown in the picture on the right. The CAR receptors would be the red ones. It allows the T cell to directly bind to surface proteins on cancer cells. Importantly, CARs bypass the normal requirement for antigen presentation through MHC molecules, giving T cells a more direct and reliable way to identify cancer cells. How are these CAR T cells made? The manufacturing process behind CAR T cells is complex. It begins with leukapheresis, where T cells are collected from the patient's blood. These cells are then genetically modified, typically using viral vectors. However, more recently, CRISPR is being used to modify the cells. After modification, the cells are expanded in a bioreactor until hundreds of millions of engineered cells are produced. Before infusion, the patient receives a brief course of lymphodepleting chemotherapy in order to reduce the T cells in their body to create space for the CAR-T cells to engraft and proliferate. After infusion, CAR-T cells expand further inside the body and begin their anti-tumor activity. Currently, CAR-T cells have revolutionized the treatment of certain blood cancers. In some forms of leukemia and lymphoma, remission rates exceed 80%, and many patients who had no remaining treatment options available have achieved durable and long-term treatment. These results demonstrate the remarkable potential of immune cell engineering. However, CRT therapy also has important limitations. Solid tumors remain much more challenging due to their microenvironment and physical barriers. And in addition, CRT therapy can produce certain toxicities. One major toxicity is CRS, or cytokine release syndrome, where CAR-T cells activate and release cytokines, which then also stimulate other immune cells to release even more cytokines. This creates a runaway inflammatory loop. Clinically, CRS can cause high fever, low blood pressure, and respiratory difficulty. Currently, the CRS syndrome is being managed with drugs that block interleukin-6 signaling, along with supportive care and sometimes uh, corticosteroids. Another notable toxicity is neurotoxicity, also known as ICANS. This occurs when inflammatory cytokines increase their permeability of the blood-brain barrier, allowing cytokines and occasionally immune cells to enter the central nervous system. This can temporarily impair the neurological function, leading to confusion, difficulty with speech, or in severe cases, seizures. However, fortunately, ICANS is a very rare case among patients. So what does the future of immune cell engineering look like? Research in this field is moving very rapidly. Scientists are developing CRISPR-edited immune cells that can incorporate multiple gene modifications to improve safety, precision, and persistence. Another major direction is universal or off-the-shelf CRT cells, which could be manufactured from healthy donors and used in patients without needing to engineer each person's own cells. In addition, CAR NK cells and CAR macrophages are also being explored as strategies to better target solid tumors, as I mentioned before. Beyond oncology, engineering immune cells have potential applications in a wide range of diseases. Regulatory T cells can be engineered to better control autoimmune diseases, such as type 1 diabetes or inflammatory bowel diseases. Engineered immune cells may also help treat chronic viral infections or promote immune tolerance after after organ transplantation. Thank you for listening, and that is all for my presentation.